Hello, I'm Leonie and I'm from Run4Rhinos. Thank you for participating in our webinar series of 2021. Before we begin, I would like to encourage each and every one of you in making a donation or buying some of our merchandise to help us support and raise funds for Wild Hearts Rehab Center which rear and raise abandoned and orphaned wildlife species and rhino calves. First on our list is Dr. Johan Stale. Dr. Johan is a senior lecturer
Hi, Leonie, we can't hear you anymore. I think you cut off.
I think Leonie's, she just sent a message that their power went off. I will just proceed to introduce Dr. Johan Staal. He's a senior lecturer at the Veterinary Pathology Department at the Faculty of Veterinary Science, qualified in 2001, and he's since been involved with research dealing with hemioprotozoal disease in African antelope, and he completed his MSc on telerosis in roan and sable antelope. He received his diagnostic pathology training at the Faculty of Veterinary Science, and he's since been a part of a team of pathologists occupied with pre- and postgraduate pathology training, routine diagnostics, and research with a special focus and interest in African wildlife disease. Dr. Staal, thank you so much for your time that I know you give willingly and free for this project. And just before you start, just another appeal for people to donate. The link is in the chat. We would also ask all the participants to put their questions in the Q&A section for Dr. Staal to answer when we are through. Dr. Staal, over to you. You can share your screen.
Thanks for that introduction. I hope you guys can hear me. I just want to see if I can.
The screen is up. Everything is perfect. We hear you.
Everything is working. Okay, yeah. Good. Just see if I can get. Okay, no matter. Anyway, let's continue. Yeah, thanks for the invitation to come and talk about this. Leonie has told me that the audience today is going to be pretty mixed. So it's going to be a bit challenging to, to stay at the level where everybody will be able to feel comfortable in understanding what goes on. But it's only really one way of doing it. Hopefully the message or the basic messages that I want to convey about internal parasites in a rhino can come through in a talk. Let's just go through a couple of facts about GIT or gastrointestinal endoparasites in a rhino. It's very important to understand that under natural extensive conditions, and many of you may have seen this before, there's a very high diversity of parasites in the gastrointestinal tract of rhino, and also quite high, the individual numbers of these various species are relatively kept within certain numbers. A total number is quite high, but it's very important to realize that there's a very high diversity of parasites. So this means that there's actually quite a delicate metazoan biotic relationship, and I think anyone that's got a good idea of how a healthy ecosystem works should understand the intestinal tract of any animal, and rhino is no exception. There is a very delicate balance of a bacterial metazoan, like in this case helminth, organisms that lives very happily among each other. And that delicate balance, if it's disrupted, can cause some issues. And we're going to talk about some of those a bit later. And those issues can certainly happen under intensive and captive conditions. So in captive conditions, it is important to realize that the normal life cycles of the various parasites are seriously disrupted. However, certain life cycles remain intact, especially the direct life cycles, and it can result in overrepresentation of only a small number of specific parasites. So you can get a very low diversity, but each of those numbers can be quite high, and then you can have a so-called this metazoan dysbiosis. Okay, so that is the importance of just this is very important to understand those two concepts firstly. It's also important to realize that healthy rhinos, so rhinos that are non-stressed, have quite a high parasitic resilience, hence they can actually carry so many parasites without showing ill effect. It's a very difficult concept for veterinarians out there to grasp or understand. We're used to dealing with domesticated animals where if we see there's worms, those worms got to die. Okay, so it's important to realize that with wildlife species, and the rhino is one of them, that you need to actually take care sometimes under certain conditions. You need to take care of those parasites, since they actually create a very healthy environment in the GIT. But then it's in the same breath, one has to also understand stressed rhinos, so animals kept under intensive conditions, animals that's relocated and so forth. They can actually show now a susceptibility to parasitic pathology. So if an animal is stressed, its immune system can be affected, and then these parasite, the relationship between the host and the parasite can then change. And then the seemingly non-pathogenic parasite can then become a problem. So there are certain instances where you have to take into account, this particular issue that can arise, and then you need to act as a veterinarian or as a rhino farmer. This is an article, a very short little article that's published by very famous parasitologists at Honestpoort about parasites in African rhinos. It's a bit of a documentation of what you can get, and this is not only on internal endoparasites, it also sort of covers a couple of other things as well, your external ticks and so forth, and flies and so forth. So it's a very short little article. You're welcome to go and find and read it. But the introduction here is essentially very important to understand, and I agree 100% with the authors in that free-living wild animals is a balance between the host and the parasites. Both have evolved together over millennia, and the presence of the parasites under these conditions is usually incidental and of limited clinical significance. Okay, so here that it's limited clinical significance under extensive conditions. Under stressful conditions, however, droughts, especially human interference such as capture or captivity, then the host immune system is suppressed and the balance can be disturbed, and parasite populations may escalate to the extent that clinical symptoms may become evident. So that little paragraph sums up what you should think and how you should think about rhino internal parasites. Right, so I'm going to briefly now quickly introduce you to these guys. They are quite interesting and scary looking beasts, sometime. But here we've got the first, this is actually not a helmet per se, it's in fact a fly, the Gyrostigma species. So Gyrostigma species is a fly. The adult is a free living fly which lays eggs essentially on the mouth parts or on the hide of the rhino, and the animal can lick these off, or eventually if it's on the lips or close to the mouth parts, these larvae will hatch and they will start to migrate in the oral cavity, essentially under the epithelial surfaces, and then molt to your second and third instar larvae into towards the esophagus. So they actually migrate in the mouth parts and the epithelial layers, and then they move towards the esophagus. It's a bit unclear from there what happens, but it's probably a very short route from there to the stomach, where they then become the final instar larvae, as you can see in this image here, and then they will inhabit the esophageal part, or the non-glandular part of the rhino's stomach, as you can see in high numbers. So it can be quite alarming when you see these and you wonder about their significance. But as I say, keep in mind, under what circumstances they're in fact quite healthy. They can be, they can cause with naked eye, and I'm going to show you some images. I think the next, no, let me just go back. I'm going to talk about it a little bit later, but they can cause to the visual eye quite significant injury to the mucosa, essentially. But histologically, you can actually see that it's actually a very shallow ulceration that is formed by these parasites, and many people think that they cause severe ulceration in rhino stomachs, and that's in fact not the case. They are hematophagous, so they survive on the tissue fluids, which is formed in response to the parasitic infection in the sites of attachment, as well as blood that is going to be leaking through. And the rhino is seemingly quite adapted to that, and we're going to talk about that a little bit later as I show you some changes that you can expect at post-mortem of these animals. In the next group of commonly seen parasites is the so-called pinworms, Oxyurus species, as you will know. In rhino, there's also a smaller one, a very tiny little one called the Propstmyria species. That's been, I don't think it has been described yet, or it may have been actually described, but they are part of the pinworm family. You find them in the colon, and again, their significance is questionable. And we certainly have not seen, found any problems associated with these. Then we've got a very large group, and guys, you will all be, as I'm going through these parasites, I can even flick back to this one. In horses, a similar one is known Gastrophilus. So you can see that we are dealing here with hindgut fermenters and very similar looking and similar classes of parasites that we find in hindgut fermenters such as horses and rhinos. All right, so here we can also, similarly to horses, find strongyle species, a big group of them, and the most common ones are the Chiloluma species, Galeria, Marscheria, and Quilonia species. Okay, so these are all different types of strongyles living in the colon, and later on I may talk about their significance, especially under intensive conditions. So these guys can become a problem under intensive conditions. All right, then another group of class of endoparasites that we see in the rhino is Anapocephala species, the tapeworms in the colon. Here we can see them, these very large white structures. They're actually quite big in a rhino, as big as your thumb essentially. They look actually quite tasty with a bit of, maybe sauteed with a bit of butter. No joking just there. Okay, so but here you can see them in association, and you can see their size in relation to the strongyles and the pinworms that is also mixed with the feces. So you can see in a normal extensive rhino, lots and lots of variation of high numbers of parasite, and you must be careful of how you interpret the rest of the changes you may find on this animal if you see these parasites. Then lastly, Gastrodiscus, which is now the trematode. So we've talked about some flies in the stomach. We've got the nematode groups, and then we've got the cestodes, and here we've got the trematodes or the flukes. Personally, I have not, this is in fact a picture not from a rhino but from a horse, but it has been described just to show you what they look like if you haven't seen them before. Personally, I haven't seen them in rhino before, so it's probably not that significant, but it has been described in the literature. So it's not just something that occurs in horses. So flukes, same to the tapeworms. Flukes have an indirect life cycle. So these guys, same as your liver fluke, as you may better know, has an intermediate host of a snail, the Bulinus species in this case. Is free-living snails in natural water sources. So that is where your Metasacaria is going to live, and that is also how the animal then get infected with the final stage here. Just back to the tapeworm stages, they also indirect life cycle. It's quite an interesting life cycle that they've got in where the intermediate stage, which is a cysticercoid, lives in vegetative mites. The mites that live in the grass and they actually feed on vegetation and so forth, they can ingest in the feces, they can ingest the eggs of these tapeworms, and then the mites can then be ingested by the rhino as they're grazing, and then they get infected with the final stage. Okay, so that's basically how the life cycles of these various parasites work. And then the roundworms are a direct life cycle, eggs on pasture and then direct intake. All right, so let's look at the veterinary considerations that we need to focus on. Firstly, it is important to know that we don't really know much about gastrointestinal physiology, and we have to really be careful about how we apply our knowledge that is based on domestic animals, on rhino, as I said earlier. There are very few primary gastrointestinal disorders, and even in these, we've got to consider what is the possible role of the helminths in them. And sand impaction, for example, even small rhino, stressed little rhino in captivity, they can really get issues with this. I'm sure some of the guys talking about hand rearing can talk you through the occurrence of this, the frequency and the treatment of these. But sand impaction can be an issue, and then again, you've got to ask yourself, what would be the issue if, could there have been sand impaction, for example, if there was a proper, healthy helminth environment? We don't know, but I mean, I think it's more to do with the environment and the stressful situations than the unnatural situation than the helminths itself. And then we can get sporadic cases of the common bacterial diseases that we know, like salmonella, for example, that's been seen in animals that drink water from sewage contaminated sources and so forth. So that is also something that we did sometimes see. A very common thing with rhino is so-called post-relocation maladaptation. And here is now an instance where a very high helminth population can act opportunistically when the animal is moved or translocated to a very different area, and that animal struggles to adapt for various reasons, for social and environmental reasons, for nutritional reasons. All those things will play a role in its adaptation success. And there certainly, if the animal do become immunosuppressed because of maladaptation, the helminths can become a contributory or a secondary problem that may cause further issues in letting the animal lose condition, which will snowball the effect. In cases like that, it may in fact be indicated to treat them for the parasites. But we're going to get later on to why you should be careful of treating. And then, as I said earlier, I'm going to get back to that. So I'm going to show you some pictures of gyrostigma, where these botflies essentially attaching are not clinically ulcerogenic. So many people think that they are. They certainly do not cause clinically deep enough ulcers to be a problem for the animal. I'm going to show you some pictures of it now. So now let's look at the helminth associated problems. So we have in intensive conditions seen that for certain species, and I suspect it may be the smaller strongyles like the Chirolima species, which migrate in the mucosa of the as part of the life cycle of the larvae, similar to your small strongyles Acetostomum species that you see in horses, they can migrate in the mucosal surface, and there they can in fact cause a little reaction, a little granulomatous reaction to the parasite, and this will actually cause not just granulomatous inflammation, but it's also going to cause colonic hyperplasia, mucosal hyperplasia, and this can cause a break in the mucosal layer, which can, in very high infestations, possibly become a problem for secondary invasion of bacteria, like we've had in outbreak situations, which I'll explain later. Then the protein energy malnutrition story that I talked about in the maladaptation, the role of the helminths that contributes secondary when animals are stressed. And then importantly, and we've seen this a couple of times, where an enterotoxemia or enteritis develop as a result of Clostridial proliferation. The cause of it may not be so apparent, but some of it is seriously has been helminth treatment has been implicated. So it stands to reason, if you have such high levels of helminths in the intestinal tract, you suddenly kill them with an antihelminthic, that will become a protein source to be utilized by the local bacteria. And Clostridia is one of the organisms that is very efficient and good at that. The problem with that is the Clostridia themselves can become a problem for the animal and cause severe enteritis and then potentially sometimes even death. Okay, back to our Gyrostigma species causing so-called ulcerations. Here we've got a couple of them pulled off their attachment sites, and you can clearly see there in the non-glandular part, there seems to be a crater-like structure here that seems quite deep. Histologically, though, the stomach wall is extremely thick, and this keratin layer is essentially a very thick layer, and this ulcer is actually quite shallow. So if you look at it histologically, it's not nearly as bad as it looks like on a macroscopic level. And I think people, veterinarians, sometimes misinterpret this. We need to kill these worms. That is a kind of thinking process. Kill them because they cause ulcers in the stomach. That is not true. So be aware of how you look at these worms, or these flies, actually, not the worms. Right, here we can see again in the non-glandular part, these so-called ulcers, which in fact is just shallow erosions in the context of the thickness of the stomach wall. They're in fact quite shallow. So they're very, very, very shallow without causing much problems. Then what I said earlier about the relationship and the reason why I think there's a quite close not necessarily commensal, but a very balanced relationship between gyrostigma and rhino is if you look at the proximal duodenum here. So you're looking at the pyloric area, and this is the proximal duodenum. And you can see there's quite a sort of a blackish gray and even light brownish discoloration of the mucosal surface. Now, histologically, looking at these areas, you will see that the villi tips are full packed with macrophages containing hemosiderin. So we said earlier that the rhino, well, what did we say? Yeah, we said that the gyrostigma is hemophagic, which means that the feces they produce in the stomach is potentially going to contain quite a bit of iron. So I think and this is only a hypothesis from my side, that the rhinos are quite adapted to actually regain the iron that's lost by the worms feeding in the stomach. By a special mechanism where they actually resorb the iron back into circulation. This potentially actually poses a potential risk for the animal, since diets that are high in iron may actually cause iron intoxication, which has been reported in rhino if they take up too much iron. You know, since they preserve iron because they're losing it from the worms. Right, so one has to actually be cautious about high iron diets in rhino because of this particular relationship. This is the colonic mucosal surface, which shows some abnormality, and this is the multifocal little pale areas that's raised in the colonic mucosa represents mucosal hyperplasia and underlying granulomatous inflammation in the colon, and this is the result of tiny little migrating larvae from the Chilolema species, I suspect. On a histological level, you can see here on the right here, there's some sort of what the colonic epithelium is supposed to look like. Here you can see quite a very marked goblet cell hyperplasia taking place, and then here you've got actually cross sections of the migrating parasite causing a fibrotic reaction here. But you can actually see the animal's got a very, it doesn't have an over active immune reaction to the parasite, which often causes more tissue damage than the parasite itself. So you can see there's actually it's almost like the rhino's immune system is tolerating this parasite's effect. As I said earlier, be careful of the number of these in the mucosa that can cause eventually some injury that may potentially lead to problems. And this is exactly what has happened in some instances where in intensive farming system, and this is now not farming systems where animals are kept on a non-pasture, so it's not a no grazing system. This is actually a pasture system, but it's intensive animals. So there's a very high load of parasite over deposited on the pasture and recycled all the time, which means that the animals are getting very high loads of the so-called small strong gulls of Chilolema in. And what has happened is we've associated with this, we had outbreaks of Clostridium novii myositis and mortalities, acute mortalities in the rhino. And this in fact was the reason why the vaccine RhinoVax was developed and produced and is regularly now applied in animals in intensive and semi-intensive situations to try and decrease the risk of this problem occurring. But as I said earlier, under semi-intensive systems, this is where you as a veterinarian need to probably apply your common sense in treating rather for these worms, since intensive systems is going to be a problem for this kind of issue. The exact pathogenesis for why this relationship exists is just a suspicion. It is known that anything causing mucosal injury at any place in the GIT can lead to the spores of histotoxic Clostridia settling in muscle tissues, for example. And when the circumstances arise where there's anaerobic conditions, you can have them germinate and then cause disease. So this is the relationship between the parasite and the Clostridium, which I suspect may be happening. Okay, so close to finishing off. So let's summarize quickly, and because I'm sure at some stages you got confused. Should I now deworm rhino or should I not deworm rhino? So this is the basic summary. Free-ranging rhino, you don't deworm them. All right, so this may result in, if you capture a free-ranging rhino, you immediately deworm that animal with a broad spectrum, you can cause a potential bacterial dysbiosis and in sometimes a clostridial enteritis, okay? So the examples here are felt to felt. If you're going to relocate an animal from felt to felt, you'd rather leave the animal with its parasites. He is going to just fit in nicely or better into the other side. Felt to temporary captive, also leave it. If you're going to permanently captive, you're going to sit with a captive situation here, and the best situation in these circumstances is if you're in an intensive system, you need to apply anthelmintic treatment to make sure that you don't get an overrepresentation of one of the direct life cycle nematodes like your small strong owls. Okay, so that should make sense, and then clearly also apply vaccines that's made for this purpose, like RhinoVax, for example, to make sure that the animals do not contract or do not become susceptible to this condition as well. So that's a basic situation. If you are going to, for example, put an animal for long-term in captivity, this often happens if animals are quarantined for a period before they may be exported. There's no reason and there's no rush for why you should at capture already deworm these animals. Why should you do that? It's unnecessary. So you might as well leave that animal at that stage, capture the animal, relocate it to the boma without deworming it. And then later on, as the animal has adapted now to its new diet, new environment, less stressful now, you can then start to think of deworming the animal. Okay, but you will have to look at each animal individually to see how that works. But doing all these things at once, treating them, many people believe they must give them antibiotics. I certainly will not advocate that at all, and certainly also not helminths. A big reason for also another big drive not to deworm animals when you relocate them is, remember, there's not one anthelmintic that is 100% effective. So, which means if you're going to deworm an animal before you move it to another farm, the only thing that's going to happen is you're going to drop resistant worms on that side. So there's going to be some worms that's going to survive in an animal's intestinal tract. And the only effect that you're going to gain by that is to actually cause resistant population of worms against the drug you used in that relocation. So there's really no sense, and I challenge people to try and convince me otherwise, but that is my opinion about this matter. All right, that's essentially it. So it wasn't a long story. I'm encouraging all kinds of questions. So hopefully there is some. I hope I didn't confuse you guys too much. So I think, let us see, where is this?
Hi, Johan. So there's one question. How is the vaccine RhinoVax administered?
Okay, there we go. I see the questions now. Sorry. Right, so how's the vaccine RhinoVax administered? I mean, it's like all other vaccines. It's important when you are dealing with vaccines to to remember that you need to maintain the cold chain. All of you that is, we're all familiar now, that everybody's familiar with vaccines, with COVID, you know, this global pandemic. And there's been lots of talks in the media about how the vaccines had to be kept cold and so forth. That's a very, very important matter. These vaccines, not all of them, but most of them requires a cold chain to be kept. And even if a batch has not been expired, if at some stage that vaccine got a bit of heat, it may become inactivated and that vaccine will not work. So that's the first important part of understanding how to administer a vaccine, is that to keep the cold chain. You keep that cold chain up until you're exactly next to the animal where you're going to vaccinate. And it's simply just you know, pulling up the vaccine, mixing it. If it's not already in a fluid substrate, you can then just administer it subcutaneously. So this is a subcutaneous vaccine. Many farmers may even opt for vaccinating. This vaccine is used to other species as well. For the histotoxic Clostridial complex that's in there, as well as some other antigens like anthrax. So there's quite a multi-disease protection in this particular vaccine. So other farmers, you know, if the animal doesn't need to necessarily be immobilized, some farmers will actually use this themselves by remote dart administration, and that's perfectly sound as well, as long as you make sure that your darts are kept quite cold the whole time. You don't put it, you put it in your dart when you're ready to dart the animal. You don't put it at home and leave it in your bucky, and then when the animal is 2 hours later available, that's going to be a problem. Keep it cold on ice the whole time, and just before you want to dart an animal with a dropout remote darting system, you take it out of the ice. Okay, so that's important to remember. Only one question.
You gave a very comprehensive lecture. Is there any other closing remarks from your side?
No, from my side, that's it, as you can see.
Hi, everyone. I'm Alex. I'm just quickly going to take over for Leonie and conclude for us. I just want to say thank you so much, Dr. Stale. That was extremely interesting and I enjoyed it a lot. And then I just want to also thank all the participants. I hope you enjoyed it as much as I did. And then I just want to ask you all again to consider donating by scanning the QR code on your screen now. All donations go to Wild Hearts, which is an amazing organization. They don't only take care of orphaned rhinos, but a wide variety of animals, wild animals. And if you would like to purchase any of our merchandise or plushies that we sell, all the profits also go to Wild Hearts. And if you are interested in that, you probably saw the video at the beginning with all the merchandise we have, then you can follow us on Instagram at Run4Rhinos or on Facebook at OP Run4Rhinos Fun Run. And the order forms are on our social medias. And as well, if you have any further questions, you can directly message us. And then, yeah, I want to thank everyone so much. And tomorrow we are having another session with Dr. Jacques Sedal about dehorning, which should be interesting as well. So I just want to say thank you so much for everyone for joining, and you all should have a good night.
I just want to point everybody's attention to the link in the chat. There is a CPD link. I think the first one had an error on. Please, you can try that one. And with a form, yes, it works. So with a form, you can claim your CPD. Thank you so much for attending.